Hello. Oh, hello, hello. Am I audible? Hello. Good evening. Hello. Am I audible? If yes, you can give me a thumbs, okay? Hello. Hello. Okay, very good evening to you all. So you are at the verge of examination, right? So I'm here to discuss 50 important questions that is being in your textbook and in your that is going to that possible questions in your textbook. So once again, welcome to Gosco Learning, GCC's fastest growing ethic. Okay, so here again I am Amala Anjos, your biology teacher, and as I told, today we'll be discussing with the most important MCQs. Okay, the most important MCQs of grade 12 that is going to be happened in coming 19th. Okay, that examination is on 19th, right? So you are ready to fight. Yes or no? Yes. Okay, so let's start the MCQs of chapter number 4, that is Principles of Inheritance. Okay, why this chapter? Why I have chosen this chapter is because, see, among the different units of your textbook, this unit, second unit, is a very important unit which you can score 20 marks from 3 chapters. And today we'll be discussing about the 2 chapters and its MCQs. Okay, it will surely help you to score good marks. And again, we are going into the question. Okay, so what is the first question? This is the chapter name. Okay, so what is the first question? The case of Down syndrome in humans is. This is the question. Okay, so what are the different options given here? Options are extra copy of an autosome, then extra copy of a sex chromosome, absence of an autosome, absence of a sex chromosome. To answer this question, we need to know what is that disease, Down syndrome, right? Down syndrome can happen if an extra chromosome is added to the 21st chromosome. Okay, it is if a chromosome is added to the 21st autosomal chromosome, then we can say that condition is known to be the Down syndrome. Okay, so this Down syndrome can be written as trisomy 21. Okay, Down syndrome can be written as trisomy 21. So what will be the answer? What will be the possible answer for this question? See, once again, the question, the case of Down syndrome in humans is extra copy of an autosome, extra copy of a sex chromosome, absence of an autosome, absence of a sex chromosome. So we can avoid these two answers since it is not dealing with sex chromosome. Okay, so next one, the possible answers are extra copy of an autosome and absence of autosome, right? So we have already studied that it is trisomy 21. 25th first autosome is added with one more chromosome and it forms the trisomy 21, right? So we can say the answer is extra copy of an autosome. Okay, extra copy of an autosome leads to the formation or leads to the condition called Down syndrome, and we can say Down syndrome can leads to different conditions where sperm is retarded. The entire growth of the human being is in a retarded condition. Okay, you have to study the features of that disease too. Okay, clear? Fine. So can we move to the second question? Can we move to the second question? Yes or no? Okay, so let's move into the second question. What is the second question? In sickle anemia, at which position the amino acid gets changed? Okay, that is the question given here. So you have to study different Mendelian disorders, right? So what are the different Mendelian disorders you have to study? Is about sickle cell anemia. Okay, it is about sickle cell anemia. Then you have to study about thalassemia. Okay, then you have to study about hemophilia. Okay, then you have to study about color blindness. Okay, there are different diseases that is to be noted in your textbook. So, in order to remember the different diseases, I'll say a code for you. Okay, what is that code? C, ta, ra. Okay, different diseases we have to study. What diseases? Mendelian disorders. And this Mendelian disorders you have to study, is it an autosomal recessive disease? And also you have to study if it is a sex-linked recessive diseases. Okay, so here we have four different diseases. Among that, two is autosomal recessive. Let's see how this two can be connected. Okay, first one C, right? C, this C we can say that it is sickle cell anemia. It is sickle cell anemia. Okay, then we'll go with thalassemia. Okay, we'll go with thalassemia. Okay, then you have R and A, right? R and A. See, write this as auto. A is for autosomal. R is for recessive recessive condition. So we can say sickle cell anemia and thalassemia are autosomal recessive diseases, whereas the hemophilia and next one color blindness are sex linked recessive diseases. Okay, so sometimes you may get the question to answer it in a condition where you have to say whether this disease is autosomal recessive or sex-linked recessive. Okay, so study about the four diseases. Along with this, you have one more diseases, that is phenylketonuria. Okay, so in what is the question here? Apart from this, what is the question here? Sickle cell anemia, at which position the amino acid gets changed? Okay, so what is the condition here? Here, glutamine. Okay, here what is the condition? Glutamine for sickle cell anemia. Okay, for sickle cell anemia, the glutamine. Okay, glutamic or glutamic acid. Okay, glutamic acid. We can say glutamine. Glutamine. It is converted into valine. Okay, at which position? Sixth position. At which position? Sixth position. So, the answer of this question is sixth position. What is the answer of this question? Answer of this question is sixth position. Okay, is it clear? Clear for you? Okay, glutamic. We can see this glutamic acid. Okay, glutamic acid. Glutamic acid is converted into valine. Why it is converted into valine? The base pair which is present inside that will be mutated. Since it is mutated, the mRNA produced will be mutated. The mRNA produced will be coding for the valine. Okay, and in which position it is? It is in the sixth position. Okay, you have to study the position. Okay, clear. So next one, what is the next question? What is the next question? Okay, again we have another question. Trisomy is found in which condition? Okay, as I have mentioned before, is it in Turner syndrome? Turner syndrome, you have to remember that it is X0 condition. Okay, Turner syndrome, X, we are turning something, right? Turning. Okay, if it is turning, then complete the turn. So we can say that it is 0. Okay, X0 condition. Turner syndrome is X0 condition. Okay, then Klinefelter syndrome, we can say that it is XXY condition. XXY condition is for Klinefelter syndrome. Okay, next one, Down syndrome. We have learned Down syndrome as trisomy 21. Okay, what is trisomy 21? Trisomy 21. In the 21st autosomal chromosome, there is one additional chromosome. One extra chromosome is present at the 21st chromosome. Okay, so the answer is Down syndrome. Okay, trisomy is found in Down syndrome. Okay, clear? Okay, so next we will go with the next question. Okay, so what does this question says? Pleiotropy can be defined as. Okay, what is pleiotropy? We have learned pleiotropy, right? One gene, one gene, it is expressed in many expressions, okay, or many traits. It is expressed in many traits give rise to pleiotropy. Okay, in your textbook we have to study the example for pleiotropy is phenylketonuria. Okay, phenylketonuria. Okay, phenylketonuria is an example for pleiotropy. So what is the answer here? Here the options are when one gene control one trait. Is it the answer? No, this is not the answer. When one gene exhibit multiple traits. Yes, this one is the answer. What are the other options? When multiple genes control one trait. Okay, is that one? No, it is not the condition. When multiple genes control multiple traits. It is not the answer. So what is the answer here? The answer given here will be when one gene exhibit multiple trait. Okay, one gene which gives many traits. Example, in your NCERT textbook it is phenylketonuria. Okay, clear? Is it clear for you? Okay, so next question. We'll move to the next question. Which of the following organism has X0 chromosome in male individuals? Okay, so here it is about the sex determination in different organisms. Okay, if we take or if we look into the sex determination of different organisms, first we have to discuss about human beings. Okay, so human beings shows male heterogamy. Okay, human beings shows male heterogamy. Okay, what is actually male heterogamy? Here see the females have X chromosome, two X chromosome. Okay, whereas in the case of males, they shows XY condition, X chromosome and Y chromosome. Okay, and also the male heterogamy can be exhibited in insects. Insects also it is male heterogamy where females are XX. Okay, where females are XX and where males are X0 condition. Okay, so how the genes will be formed or how the gametes will be formed here in the case of male, it is X and Y. So the gametes formed is X and Y. Okay, in the case of females, it is XX. So the possible gametes are X and X. So the progeny is, sorry. Okay, here the gametes are X and Y. Okay, so the progenies will be forming like this. XX will be formed. Here XY will be formed. Okay, then here XX is formed and XY is formed. That a boy is formed. This is a girl. Okay, this is a boy. And this is a girl. Okay, this is a boy. That means 50% of, there is a chance of getting 50% of boy and 50% as a girl. Okay, so this is the male or sex determination in human beings. Whereas in the case of insects, you have two conditions here. Females are XX and males are X0. So the possible gametes will be XX and X0 will be, there is no gene present inside that. So we can consider to be that zero condition. X will be the possible gamete that will be transferred to the next generation. Okay, so here we can say male heterogamy is exhibited in the case of both the both the insects and in the case of the human beings. Okay, since it is exhibiting the male heterogamy, the human beings and insects shows the male heterogamy. Okay, this is very important. Male heterogamy is exhibited in human beings and in insects. Okay, whereas in the case of fowls, okay, whereas in the fowls, it is female heterogamy. Okay, it is female heterogamy. How female heterogamy is possible? See, it is represented by the Z gene and W gene. Okay, females are ZW. Okay, females are the organism which carries ZW genes, and the males are the one who carries ZZ genes. Okay, so how will be the gametes formed? The females are Z and W, males are ZZ. Okay, so what are the possible gametes? Z is possible gamete, W is the possible gamete, here Z is the another possible gamete. So the progeny is formed will be ZZ or ZW. Okay, that means if ZZ is formed, this will be males. If ZW is formed, this will be females. Okay, clear. So in the question, what is given? Which of the following organism has X06 chromosome in male individuals? Okay, here the answer is C. What is the answer here? Answer is C. Okay, clear. Got it. So next one, we have to go with the next question. In a human zygote XXY sex chromosome along with 22 pairs of autosome, what will be the sex of the individual they developing from the zygote? Okay, here what is the question? The zygote is having XXY condition. What is this XXY condition? If XXY condition is there, then we can say that person is having Klinefelter syndrome. Okay, Klinefelter's syndrome. So we know that those person will be actually male. Okay, since it is having the Y chromosome, since it is having the Y chromosomes in the person or in the sex chromosome, we can say the person will be males. Okay, the person who is possessing the XXY genes are males, but they show the feminine characters. Okay, but they show the feminine characters. Okay, like broad waist, they will have less amount or less hair growth. Okay, over their beards. Okay, they are basically males, but they have feminine characters. Okay, clear, fine. Then next question, we'll be moving with next question. What is next question? Read the question, please. Okay. Okay, what is the question given here? Mother's blood group is A and father's blood group is B and the daughter's blood group is O. What will be the blood group of other children? Okay, so what is the blood group of mother? Okay, what is the blood group of mother? What is the blood group of mother? It is clearly mentioned that it is A blood group. Okay, so father, what is the blood group of father? Father, it is also given as B. Okay, so what is the blood group of mother? A. For A blood group, the possible condition will be IA. Genotype will be IA, or the genotype will be IA small letter I. It will be in heterozygous condition or in a homozygous condition. Then the fathers will be also same like that. It will be either IB or the heterozygous condition IB and small letter I. Okay, clear? So what is the given child of a child's blood group? It is O. Okay, how it will be? See, you know that the given child is child is given, child or the child's group is given that it is O blood group. If O blood group has to come, you know that O blood group is the genotype for the O blood group is II, small letter I. Okay, so we can clearly take out the possibility of homozygous condition. We can clearly take out the condition of this homozygous condition. So we can go with the condition of IAI and IBI since it is having the smaller I in the gene. Okay, so what will be the possible gametes or possible offsprings produced rather than this? Oh, let's do it. Let's do it. So we can say IAI is mother's gene and father's gene is for IBI. Okay, father's gene is IBI. So how can we do this? IA is one gamete. We can put it in the same size of circle, right? So it is also one gamete. Okay, the next one, IB is possible gamete. I is another possible gamete, right? So we can cross this, right? The possible progenies will be IA, IB, IA, IB. Okay, then next one, IA, I. Okay, next one, IB, I. Okay, then this one here it is small letter I. So what are the possible blood groups produced here? What are the possible blood groups produced here? See, IA, IB which codes for AB blood group. Okay, IAI codes for A blood group. Okay, IBI codes for B blood group. Okay, then small letter I, it codes for O blood group. So what are the possible blood groups from this particular genotype? It is AB. See, AB is possible, A is possible, B is possible, and O is possible. In our question, it is clearly given that the daughter's genotype is O blood group, and what are the other possible genotype? That is the question. So all the genotypes are possible in this condition. So what will be the answer? Comment in the comment box. What will be the answer? The answer will be all of the above. Okay, answer will be all of the above. See, mother's blood group is A, father's blood group is B, and daughter's blood group is O. What will be the blood group of other children? All of the above is possible here. Okay, clear? Fine. So can we move to the next question? Can we move to the next question? Yes. Okay, so let's move into the next question. Read the question. Which of the following features shows the mechanism of sex determination in honeybee? Another important topic. Okay, sex determination in honeybees. How sex determination of honeybees are? Do you have any idea? Okay, so this condition or this step or the process is known as haplodiploid. Okay, haplodiploid sex determination. Okay, haplodiploid sex determination. Why? Both the conditions are present. Diploid condition is also present and haploid condition is also present. Okay, let's say in the case of females. Okay, in the case of females, the females are with 32 number of chromosomes. Okay, in the case of males, if you are looking into the case of males, males are with 16 number of chromosome. Okay, so here they are in the diploid condition and males are in a haploid condition. So we can say this is a haplodiploid sex determination. Okay, fine. So next one, see, after meiosis. Okay, the process undergo meiosis. Meiosis. Okay, the female undergoes meiosis to produce how many number of chromosome? It is 16 number of chromosome. Okay, 16 number of chromosome. The gametes are with 16 number of chromosome. Here males are produced, the gametes are actually produced by the process of mitosis. Okay, it is produced by the process of mitosis. So we can say how many number of chromosomes are possible here? 16 number of chromosomes are possible here. After mitosis, same number of chromosomes are produced, right? So when 16 number of chromosome from mother and 16 number of chromosome from father, okay, or male is fused together, there how many chromosomes are produced? 32 number of chromosomes are produced, and that individual will be leading to the formation of a female. Okay, that leads to the formation of a female. Whereas in the case of males, how males will be produced? Here 16 number of chromosomes are present, right? This 16 number of chromosome under parthenogenesis, it forms the 16 number of males. Okay, it produces the males under parthenogenesis. You know about parthenocarpy, right? What is parthenocarpy? It is the formation of fruit without fertilization. Here also what happens? It is the formation of an entire organism without the process of fertilization. Okay, clear? So under parthenogenesis, what happens? Parthenogenesis, a new male is formed. Okay, so for the male, there is no father. Okay, for the male produced, there is no father, but for the males, for the males produced, they have the grandfather. They have the grandfather, and also they have the grandson. Okay, they have the grandson. This is important. This is important. Okay, males produced will have a grandfather, and also they will have a grandson. Okay, so now let's move to the question again. Read the question. Which of the following features shows the mechanism of sex determination in honeybee? Okay, so what is the questions given here? Options given here are formed from the union of a sperm and egg develops into a male. Okay. If union of sperm and egg is formed, then it develops into a male. Is that statement correct or not? It is not correct, right? So males have half the number of chromosomes that of a female. Is that statement correct or not? Is that statement correct or not? That statement is correct. Okay. The females are diploid having 32 chromosomes. The statement is correct or not? That statement is also correct. Okay. The next statement, males have father and can produce son. Do the males have father? No, they cannot produce the sons too. They can have the grandfathers and they can have the grandsons. Okay, clear? Understood? Okay, fine. So that is all about the question. Okay, so we can go with the answers as 2 and 3. If the options you have, then both the statements are correct. Okay, fine. What is the next question given? Select the pair that is incorrect. Okay, here we have to select the pair that is incorrect. So before that, I told you, right, in the beginning of the class, there is a code. The Ra. Sitara. So sickle cell anemia, thalassemia are autosomal recessive disease. Okay, thalassemia are autosomal recessive disease. Let's see. Sickle cell anemia. First option is sickle cell anemia. It is autosomal linked. It is linked. Okay, not like it is linked. Okay, linked recessive disease. Is it correct or not? Is it correct? Yes, it is correct. So second one, it is given as hemophilia. Hemophilia is autosomal linked recessive trait. Okay. Then color blindness, sex linked recessive trait. Thalassemia, autosomal linked recessive trait. So what is the incorrect answer here? The incorrect option given here is okay, the incorrect option given here is which one is the incorrect option? Sickle cell anemia, it is autosomal linked recessive trait. Okay, then thalassemia, it is also autosomal linked recessive trait. The rest of the two, you can study that color blindness and hemophilia. Color blindness is also a sex linked recessive trait. Okay, then the next one it is hemophilia. It is autosomal recessive trait. It is given. That option is correct. So the correct answer is second one. Okay, which pair is incorrect? Hemophilia, autosomal linked recessive trait is the incorrect statement. Okay, is it Clear? Is it clear for you? Yes or no? Okay. So next question. What is the next question? An example of human trait where a single gene can exhibit multiple phenotype expression is given. Is given as phenylketonuria, cystic fibrosis, thalassemia, and hemophilia. We have discussed this in the previous question, right? Yes or no? Is or no? Okay, so what is the answer here? The answer here is phenylketonuria. Okay, how this is working? See, there is an enzyme. Okay, there is an enzyme known as phenylalanine. Phenylalanine hydroxylase. Okay, we have an enzyme known as phenylalanine hydroxylase. What is the role of this enzyme? Here this enzyme have to convert the phenylalanine. Okay, enzyme. The role of enzyme is to convert phenylalanine into tyrosine. Okay, those person which is having this phenylketonuria, what happens? This conversion won't be possible. Okay, as a result, what happens? This phenyl pyruvic, it forms the phenyl pyruvic acid. Pyruvic acid. And this phenyl pyruvic acid, it is then get deposited in brain. Okay, it gets deposited inside the brain. So, if it is deposited inside the brain, it leads to a disease called phenylketonuria. Okay, clear? Real function of enzyme called phenylalanine hydroxylase is to convert phenylalanine to tyrosine. If this enzyme is absent or if there is any issues with this enzyme, what happens? Phenylalanine cannot be converted into tyrosine. Okay, so we can say it is tyrosine. Okay, okay, let be like that. Okay, so we can say phenyl pyruvic acid. It is then accumulated inside the brain, which may leads to the disease called phenylketonuria. Okay, is it clear for you? Is it clear? So, what is the answer? The trait. It is known as the process, or it is known as pleiotrophy. Okay, pleiotrophy, where one gene, a single gene, exhibits different phenotype. Okay, clear? If it is clear, then you can give me thumbs. Okay, fine. So let's move into the next question. Life cycle of Drosophila. Life cycle of Drosophila. Drosophila is completed in. Okay, life cycle of Drosophila is completed in. In your textbook, it is clearly mentioned that there are 14 days. Okay, but if we refer some other sources, we can say that it is 21 days. So, since your NCERT textbook says that it is 2 weeks, clearly 2 weeks, we can go with this option. What is the option? 14 days. Okay, 14 days or 2 weeks. Okay, clear? Fine. Okay, so next we will be moving with next question. What is next question? Which one of the gene pair is expected to give a ratio of 1:1:1:1:1 in the progeny of Mendelian hybrid? Okay, so what is the question given here? Capital A, small a, capital B, small b, capital A, small b, capital B, small b. It is not possible. Okay, the genotype given itself is a wrong condition. Okay, so we cannot go with that one. Okay, so what is next option? A, B, B with A. We will let's do that. A, B is crossed with capital A, capital B, small b. The ratio should be 1:1:1:1. Let's see whether it is possible or. Okay, so the gamete produced here is AB. Here, what is the gamete produced? AB is one gamete produced. AB is another gamete produced. AB is another gamete produced. Small letter A and small letter B is another gamete produced. Okay, so what will be the possible answers? Capital A, capital A, capital B will be one progeny. Then next progeny, capital A, capital B, small b. Okay. Next one, capital A, small a, capital B, capital B. Last one, capital A, small a, capital B, small b. Okay, here the look into the progenies produced. There is this will be having same phenotype. See, capital A is present, capital A is present, capital A is present, then capital B, it will give same phenotype. Here also it will give the same phenotype. Here also it will give same phenotype. Okay, so it is not expected to do with this one, this particular condition. So let's do another condition. What is the third option? It is capital A, small b, small a, then it is with question. In the question it is given as capital A, capital B, small b. It is crossed with small a, small b, small b. Okay, so this is the question. So what are the possible gametic genotype here? Here it is small a, capital B is another gamete. Then small b is another gamete. Okay, then you can find the gamete for this one is always small a and small b. Okay, so what will be the progenies produced? Capital A, it can be combined together to form capital A, capital B, small b. Okay, then this small b. Look into the genotypes. Okay, look into the genotypes. Every genotype is that different that of one. See, capital A, capital B. Here it one is capital A, small b. Capital A, capital small b, small b. Here it is small b, small b. All the genotypes are different. Okay, here all the genotype for the cross is different. So we can say the ratio produced here is 1 is to 1 is to 1 is to 1. Okay, so what is the answer for this question? The answer is third one. One is the answer. Okay, so fourth one, it is clear that only possible genotype is for the gametes is AB and small ab. Okay, so if we cross it, we'll get one product or one progeny, that is capital A, small a, capital B, small b. It also does not match with the question, so we can go with which option? Third option. Okay, clear? Fine. Is it clear? Clear? Okay. Next question. Okay, we'll go to the next question. In Pisum sativum, the flower color may be violet or white. Okay, what is the color of Pisum sativum? It is violet and white. What offspring in a cross of capital V small v into small v would be expected to be violet? Okay, that is the question. What is the condition here? It should be violet and the cross should be this one. Okay, let's do the cross. Capital V, it is cross small letter v. So what are the possible gametes? Capital V and small v. Okay, so this capital V is actually violet. Okay, the small v is actually white. Okay, so if the cross is done, we will get capital V and small v where the color is violet. Okay, where the color is violet. Okay, fine. So what is the answer? All the progenies produced will be violet. So the answer is 100%. Okay, we'll go with the option 4. Okay, next question. Read the next question. The cross between two snapdragon heterozygous for flower color bearing different colored flowers would be, as you know, incomplete dominance, right? In incomplete dominance, red flower, it is crossed with white flower. Okay, red flower, it is crossed with white flower. The genotype is capital R, it is crossed with small r. Gametes produced here is capital R and small r. When it is crossed together, we get F1 progeny and it is pink in color. Okay, the heterozygous condition, it is pink in color. So we can say F1 progeny is crossed to get F1. F1 in on selfing F1, we get F2 progeny, that is capital small r is crossed with capital R, small r. Okay, when we do the cross, and we gametes are small capital R, small r, capital R, small r, Okay, then the progeny is it is capital R, capital R. Here it is capital R, small r. Then next one it is capital R, small r. Last one it is small r and small r. Where you can find capital R and capital R both, if both genotypes are coming together, then we can say it as red in color. If this one is coming together, heterozygous, if capital R and small r in heterozygous condition, we can say that it is a pink flower. Okay. So if small letter r is present, we can say that it is white flowers. Okay. So we can say the option as, see, 25% is red, then 50% is pink, and 25% is white. Okay, 25% is pure dominant, 20% is in heterozygous condition, and the rest of the 25% is white in color. So altogether 100%. Okay, so we can say the answer is this one, B, option B, option 2. Okay, it is having 25% red, 50% is pink, and 25% of white. Okay, is it clear? Clear for you? Okay, so we'll be moving to the next question. In a dihybrid Mendelian cross, okay, in a dihybrid Mendelian cross, garden people heterozygous for violet flowers. Okay, so we can say heterozygous for violet flowers means it can be written like this, capital V. You have to write the genotype with the question. Okay, capital V is crossed with capital V, it is crossed with small, not capital V and small v is the genotype. Okay, where heterozygous flowers are crossed with homozygous white flowers. So, what is the white flowers? It is always small v, small v. Okay, and with wrinkled seeds. Okay, what is the seed condition? The seed is in a wrinkled condition. Okay, so let's say the wrinkle condition will be smaller, smaller. Here also the wrinkle condition will be the small, smaller. Okay, so what are the possible? What is the possible answer? Capital V, small v. It is crossed with smaller, smaller. And here we can say the small v, smaller, smaller. The possible genotype. Okay, for the gamete, capital V, smaller, then small v, smaller. Okay, then here it is small v and small r is present. Okay, so what will be the answer? Capital V, capital V and small r is given, and you can find here small v, small r is present. Okay, so we can say the answer will be 1 is to 1. So the in the options it is as 1 is to 1 is to 1 is to 1. 1 is to 1 is to 1 is to 1 is to 1. So that the answer is 1 is to 1 is to 1 is to 1. Okay, clear? We can go with the answer 1 is to 1 is to 1 is to 1 is to 1. Okay, so next question is for question. Okay, you have to go with assertion, reasoning, questions. Okay, just read the options that is given in the question. Okay, so what are the different directions given here? This is actually given in your all the question paper, right? In all the question paper, you will get this one. In the following question, a statement of assertion is followed by the statement of reason. Mark the correct choice as. Okay, both assertion and reason are true, and reason is the correct explanation of assertion. That is always for option 1. Option B, what is option B? Both assertion and reason are true, but reason is not the correct explanation of assertion. Third option, it is assertion is true, but reason is false. Fourth option, it is both assertion and reason are false. Okay, these are the different statement that is given. Let's go into the question. Okay, what is the question? Assertion is good example of multiple allelism is ABO blood grouping system. Okay, that is correct. Fine, right? Multiple allelism we can go with example as ABO blood group since it is having three different genes, right? It has three different genes for A blood group, B and O blood group. Okay, so IA, IB and small letter I is the gene which is coding for the three different codes. Okay, or 3D, what we can say, blood grouping. Okay, then the reason given here is when IA and IB alleles are present together, when they are present together in the ABO system or ABO blood group system, the both express in their own types of sugar. Okay, they can express in their own types of sugar, but actually what is multiple allelism? What is multiple allelism? If you want to say that multiple alleles have to expressed. Okay, so we can go with the option 2. Okay, the answer here for this question is option 2. What is assertion and true are assertion is true, reason is true, but assertion is not the correct explanation for the reason. Okay, so we can go with the option. What we can go with? We can go with the option 2. Okay, next let's see the question. In mirabilis jalapa, okay, you know this mirabilis jalapa, right? In incomplete dominance. In mirabilis jalapa, selfing of F1 pink flowers plant produces same phenotype and genotypic ratio, right? Yes, it is same. See, pink 1 is to 2 is to 1. So we can say the phenotypic and genotypic ratio for the mirabilis is correct. The assertion is a true statement. Okay, let's see the reason. Flower color genes show incomplete dominance. Yeah, that reason is also true, and reason is correct explanation for the assertion. Reason is correct explanation for the assertion. So we can go with option. What option we can go with? We can go with option A. Okay, option A is the correct answer. Okay. So next question. What is next question? By means of dihybrid clause, okay, by means of dihybrid clause, the law of independent assortment can be studied. Okay, is it correct? Is it correct? Yes, it is correct. Why it is correct? See, we have three different laws, right? First one, it is law of dominance. Okay, we have studied about law of dominance, right? Second one, we have studied with law of segregation. Okay, we have studied with law of segregation, and third one is law of independent clause. First two laws, that is law of dominance and law of segregation. Law of dominance and law of segregation is monohybrid clause. Okay, whereas the case in third law of independent assortment, the law of independent assortment, it is with dihybrid clause. It is always studying about dihybrid clause. Okay, it always goes with dihybrid clause. Understood? So the option A can be true. Okay, that statement, the reason assertion is a true reason. The law of independent assortment is applicable only linked genes. Okay, is a true statement. Can we study a dihybrid clause where when the genes are linked together? No, we cannot study when the two genes are linked together. The dihybrid cross cannot, or law of independent assortment cannot be studied with the linked genes. Okay, so what is the answer for this question? The recent statement is false. Okay, this statement is correct. Okay, one statement is correct, then the other statement is false. We can say we can go with option 3, that is reason assertion is true and reason is false. Okay, assertion is true and reason is false. Okay, then what is next question? Okay, in case of incomplete linkage, linked genes show new combination along with parental recombination. This here we are discussing with linkage and recombination. Okay, so here see what is the question given here. In case of incomplete linkage, there is no complete linkage over the different genes present in the different chromosomes. Linked genes show new combination along with parental combination. Okay, so the linked genes which is present inside the chromosome can show the parental combinations and also new combination. That statement is a statement. This statement is a true statement. Okay, that means assertion is a true statement. Now let's see, look into the reason. In case of incomplete linkage, linked genes are separated by crossing over. While we have studied the linkage and crossing over, we have learned that when the genes are linked, the linked genes can be crossed over. Okay, so which kind of genes can be crossing over? Incomplete linkage, we can say the linked genes are separated by crossing over. That means this statement is also a true statement. Okay, this statement is also a true statement. So we can say both assertion and reason are true, and reason is the correct explanation for the assertion. Okay, fine, clear. Okay, so next question. The females are diploid having 32 chromosome in honeybee. We have discussed already, right? So in honeybee there are 32 chromosomes. The statement is correct. Assertion is correct. Okay. Then it is saying about male. It is not saying about coming to the male part. So we can say the male are haploid having 16 chromosomes in honeybee. That statement is also correct. But we cannot link it with this first statement, right? So we can say assertion and reason are true, but reason is not the correct explanation for the assertion. Okay, clear? Is it clear for you? If yes, you can give me thumbs. Okay. So what is next question? Most of experiments regarding sex determination were done on Drosophila. Do you know that it is correct or not? Okay. See, most of experiments regarding the sex determination were done in Drosophila. Why it is done on Drosophila? See, I have told you in the case of Drosophila, the lifespan is only about 2 weeks. Okay, so within 2 weeks, we'll get the result. We don't want to go with a long gaps. Okay, to do an experiment, we need a certain time, right? To get an experiment, to do a particular experiment, we have to get the result. Okay, so to get the result, it is easy with us. No, so we can say assertion and reason are true, but reason is not correct explanation for the assertion. Okay, is it clear for you? Okay. So these are the different important questions that I have picked from this chapter. That is chapter number 4, Principles of Inheritance. Okay, fine. So let's go for the next chapter. Let's go for the next chapter. That is, which chapter? Okay, chapter number 5. That is molecular basis of inheritance. Okay, molecular basis of inheritance. Then one question is here. Okay, what is this question? In a DNA strand, the nucleotides are linked together by. Okay, how nucleotides linked together by phosphodiester bond. Okay, it can be textbook, the first detailing about the DNA. Before detailing, it is given about the number of base pairs present in human being. Number of base pairs present in human being is 3.3 into 10 raised. So next example in the textbook, it is given with it is about Escherichia coli. E. coli. In E. coli, it is 4.6 into 10 raised to 6. Okay, 4.6 into 10 raised to 6 in E. coli. In human beings, this human beings. Okay, then you have to study about bacteriophage lambda. One more bacteriophage you have to study, that is about psi. Okay, so here the answer will be 48502. Okay, 48502 base pairs are present in lambda, bacteriophage lambda. Okay, so we'll go with next question. What is next question? The component of nucleoside. So if you are going with nucleoside, we have studied that nucleoside is having a pentose sugar, and that sugar pentose sugar is present. Okay, there is a pentose sugar. Pentose sugar along with pentose sugar along with the nitrogen base. Different nitrogen base. Possible nitrogen base are adenine, thymine, guanine, cytosine in the case of DNA. Okay, if it is in RNA, adenine, then thymine is replaced by uracil, guanine and cytosine. Okay, it is present in RNA. So, if pentose and nitrogen base comes together, then we will call it as a nucleoside. What is this? This is a nucleoside. Okay, this is a nucleoside where, so what will be the answer? Pentose group will be the answer. Okay. The component of nucleoside are pentose sugar and phosphate group. Okay. Clear? So what is next question? Nucleotide. Okay. The question is about nucleotide. What is the nucleotide? Okay. What is nucleotide? See, you know nucleoside, right? Nucleoside, you know, right? Nucleoside, it is pentose sugar. We call it as a nucleotide. So what is the correct option here? Pentose sugar, nitrogen base, phosphate group. Pentose sugar, nitrogen base, phosphate group, none. Okay, so the option is A. Option A is the correct answer. What is the correct option? The correct option is pentose sugar, nitrogen base and phosphate group. Okay, so nucleic acid. Next question, nucleic acid. So first you have studied about nucleoside, right? Right, my dears? It is about nucleoside. Okay, so next one it is about if nucleotide. Nucleoside, once nucleosides are formed, if phosphate group is added, then it forms nor number of nucleotides come together or polymer of nucleotide comes together to form the nucleic acid. Okay, nucleic acid. So what is the option here? Nucleic acid is a polymer of nucleotide. Okay, option D is the correct. Pairing is taking place. Okay, adenine and thymine is always paired. Okay, next one, guanine and cytosine, right? Guanine and cytosine, it is paired, it is also paired. Adenine pairs with thymine, hydrogen bond. Option is option B. Okay, so what is next option? What is next option? Cytosine pairs with guanine. Cytosine pairs, sorry, next question. Cytosine pairs with guanine with how many bonds? With 3 hydrogen bonds. That is option A is correct answer. Okay, clear? Is it clear for you? Okay, so next question. Central dogma states that genetic information flows from how the central dogma can be written? Okay, by the process of transcription, what is formed? RNA. Okay, by the process of RNA, by the process of translation. Translation, what is produced? Protein is produced. Okay, protein is produced. Protein is produced. And this DNA by the process of replication. By the process of replication, what is formed? The DNA itself is formed. Okay, so this is the central dogma. So we can say the genetic information flows from DNA to RNA, RNA to protein. Okay, option A is the correct answer. Option A is the correct answer. Okay, so what is next question? Next question, find the correct statement from the below. Okay, find the correct statement from the below. One second. Okay, so what is the statement given here? In prokaryotes, DNA being positively charged. Okay, what is the charge of DNA? DNA here. In the statement, it is given as DNA being positively charged is held by some negatively charged proteins to form nucleoid. Is that statement correct? Is the DNA positively charged? Since phosphate group is present, what we can say? DNA is negatively charged. Okay, DNA is negatively charged. So that statement is false. Okay, that statement is false. Next statement, what is the next statement? In eukaryotes, there is a set of negatively charged proteins called histones. Okay, this statement is correct. You know about that, right? If this is DNA, this DNA, highly negatively charged DNA. Okay, if this is negatively charged DNA, okay, if this is negatively charged DNA, histone proteins, histone octamers, okay, histone octamers are present to wrap this DNA. They are positively charged. Histone octamers are positively charged. Why they are positively charged? Due to the presence of lysine and arginine. Okay, due to the presence of lysine and arginine, histones are positively charged, and histones, the DNA molecules will be wrapped around the positively charged. Okay, wrapped around the positively charged structure to form what is forming? Nucleosome will be formed. 200 base pairs. How many base pairs it has? 200 base pairs. From nucleosome, what is formed? A structure called chromatin will be formed. Okay, a structure called chromatin will be formed. Then chromatin fibers will be formed. Okay, chromatin fibers will be formed. From this chromatin fibers, it again wrapped together to form the which is present in the metaphase, that is the chromosome. Okay, so the negatively charged DNA, it is wrapped around the positively charged histone molecules, that is histone is having lysine and arginine, so it is positively charged, forms the nucleosome. Nucleosome is having 200 base, important, okay, 200 base pairs. It forms the chromatin. Chromatin forms the chromatin fibers. Chromatin fibers forms the chromosome, and this chromosome can be viewed only at the time of cell division, that is metaphase. Okay, it can be seen in the which phase? Metaphase. Okay, so what is the next option? Histones are rich in lysine and arginine residues. Okay, so we can say that is correct. This is correct option. See, the option B is correct or not? In eukaryotes, there is a set of negatively charged protein called histone. Is that negatively charged? No, it is not negatively charged. It is positively charged. So that statement is B is wrong. C is correct. D, 6 molecules of histone with DNA forms nucleosome. For histone is an octamer. So it contains 8. Okay, histone is an octamer. So it contains 8 molecules, not 6 molecules. Okay, fine. Got it? Understood? Clear? Okay, so what is the answer? C is the correct option. Okay, so as I have mentioned, this is another question. See, what is the question? The nucleosome has how many base pairs? 150 base pairs, 250 base pairs. The correct answer is 200 base pairs. Okay, so what is next question? Which of the following statement is not correct? Read the question. DNA is positively charged. Yes, DNA is positively charged. That is a correct statement. Okay, histone proteins are positively charged. Like yes, that is also, sorry, DNA is positively charged. Is that statement correct? No, that statement is not correct. Okay, histone proteins are positively charged. Yes, it is positively charged. Histone proteins are rich in lysine and arginine. That statement is also correct. Okay, next question is about euchromatin. Okay, do you know what is euchromatin? Yes, you have to study about both the structures, that is. Okay, euchromatic and heterochromatic is transcriptionally active. They are transcriptionally active. Okay, so in the case of heterochromatin, what is the condition for heterochromatin? What is the condition for heterochromatin? They are tightly packed. Okay, they are tightly packed. Since it is tightly packed, we can say they are transcriptionally inactive. Okay, they are transcriptionally inactive. Okay, so we can go with the statement euchromatin is transcriptionally active. That statement is also a correct statement. That statement is also a correct statement. So what is the first statement? DNA is positively charged. We can go with that option. Okay, the correct answer for this question is DNA is positively charged. That is a wrong statement. What is the correct statement? DNA is negatively charged. Okay. So next question. Okay. So the promoter site and terminator site for a transcription are located at. Okay. So in order to know this, you have to know the transcriptional unit. Okay. If you are looking into the transcriptional unit, you have a promoter. Okay. Then you have a terminator. In between promoter and terminator, what is present? What is present? There is the structural genes. Okay. So these are the So this is promoter. Okay, what is this? This is terminator. Okay, so we can say this will be always a coding strand. Okay, this strand is a coding strand. Okay, since it's coding strand, this will be a template strand. This will be a, okay, I'm writing here. This will be a template strand or non-coding strand. Both we can say. Okay, template strand and or non-coding strand. Okay, and always think that the transcription takes place in which direction? The direction of the synthesis is 5 prime, 3 prime. Okay, so this will be 3 prime, 5 prime. Okay, this will be 3 prime, 5 prime. Okay, this will be 3 prime, 5 prime, and whereas this one will be 5 prime, 3 prime. Okay, and I said that this is structural genes. This is structural genes flanking between the promoter and the terminator. Okay, so what is the question here? We have to study this transcriptional unit, and also you have to study the two terms based on the in reference to the coding strand. Okay, see the coding strand, if the position 5 prime position of the coding strand is considered to be the upstream position. Okay, with regard to the coding strand, we are saying, okay, the structural genes flanking over the 5 prime direction in regarding with the coding strand is known as upstream genes. Okay, then we have the another term which is present over the 3 prime region. It is known as downstream. Downstream genes, upstream genes and downstream genes. Okay, upstream and downstream. So this term also you have to study. So what is the condition here? The promoter and terminator site for transcription are located at first option 3 prime downstream end and 5 prime upstream end and respectively of transcriptional unit. Okay, then what is next? 5 prime up only the upstream end, 3 prime downstream end. So the correct option which we can take from this question is option B. Okay, 5 prime it is the upstream and 3 prime it is the downstream. Okay, clear? It clearly in your textbook it is given that it is with a reference of coding strand. Okay, clear? Let's move to the next question. Okay, let's move to the next question. What is this question? With regard to mature mRNA in eukaryotes, which of the following is true? Okay, so we need to know about mRNA that is produced after transcription in which organism? In the case of eukaryotes. Okay, so you know in eukaryotes, the mRNA produced from the DNA. Okay, the mRNA produced in the DNA will be in the form of heterogeneous RNA. Okay, heterogeneous RNA will be formed. Heterogeneous RNA will be formed. This heterogeneous RNA, why it is known as heterogeneous RNA? The heterogeneous RNA will be having the exons and the introns. Okay, all the introns will be removed and the exons will be combined together. Okay, exons will be combined together by the process of splicing. Okay, so we can say first it was hnRNA. Okay, this hnRNA, heterogeneous RNA is transformed to form the mRNA. Okay, hnRNA, it is converted into mRNA. So we can say with regard to mature mRNA in eukaryotes, which of the following is true? Which statement is true? Exons or introns do not appear in the mature RNA. Okay, that statement will. Can we go with that statement? Let's see. Exons appears but introns do not appear in the mature RNA. This statement is correct. Exons appear but introns do not appear in the mature RNA. Then third option, intron appear but exons do not appear in the mature RNA. This is not the correct option. Both exons or introns appear in the mature RNA. This is also a false option. First option, let's see, exons and introns do not appear in the mature RNA. This is also wrong. Why? Because in the mature RNA, we can see the exons. Exons combine together to form the mature RNA. So, what is the correct option? Exons appears, but introns do not appear in the mature mRNA. That is the correct option. We'll go with option number B. Okay. What is next question? If the number of base pairs in a double-stranded DNA is 200, okay, the number of base pairs which is present in DNA is 200. That is adenine pairs with thymine and guanine pairs with cytosine. So, number of adenine is given here. What is the number of adenine? Number of adenine is 60. Okay, if adenine is 60, the number of thymine will be also 60. Altogether, adenine and thymine will be 60 plus 60, that is 120. Okay, so total you got, that is 200 base pairs, right? 200 base pairs. So what is the question? What is the number of guanine? Easy, right? 200 minus 120, it will be 80. Okay, it will be 80. So half of this 80 will be, okay, half of this 80 will be guanine, that is 40, and half of the 80 will be the cytosine. So we can go with the option number 40, that is option number B. Okay, clear? Okay. So what is next question? What is next question? Please read the question. Okay, so what is next question? Next question given here. Discontinuous synthesis of DNA occur one strand on one strand. Discontinuous strand. Okay. So when we are discussing with the strands, it is having two different strands, right? One strand will be having continuous synthesis and the next other strand will be having discontinuous strand. Why? That is the question. Okay. A DNA molecule being synthesized is very long. This is not the correct option. Okay. Second one, DNA dependent DNA polymerase for the polymerization process. Okay, for the process of adding the nucleotide in DNA, there we need an enzyme, and that enzyme is DNA dependent DNA polymerase. Okay, so what is the role of that DNA dependent DNA polymerase? DNA dependent DNA polymerase catalyze polymerization only in one direction. It can catalyze only in which direction? 5 prime through 5 prime to 3 prime. Okay, so one will be in discontinuous synthesis. Why? Because the direction of the strand will be also in a 5 prime 3 prime direction. Okay, since it is in 5 prime 3 prime direction, the synthesis will be in a discontinuous fashion. Okay, then this option is correct. Next option, it is more efficient process. No, it is not a more efficient process. DNA ligase have to some role. The most appropriate answer is B, option B. Okay, option B is the most appropriate answer. Okay, next question. Which of the following are the functions of RNA? What is the function of RNA? You have different types of RNA, right? You have mRNA, right? You have mRNA, you have rRNA, okay, you have tRNA. So, what is the function of mRNA? mRNA is also known as messenger RNA. Okay, mRNA is also known as messenger RNA. rRNA is also known as ribosomal RNA. Okay, tRNA is also known as transfer RNA. tRNA is also known as transfer RNA. So, what is the role of messenger RNA? To give the information to next generation of next cell. Okay, ribosomal RNA which helps in the translation process. tRNA takes the amino acid from the amino acid pool to the ribosome for the process of translation. Okay, these are the different RNAs given here. What are the different options? It is a carrier of genetic information from DNA to ribosome synthesizing polypeptide. This is the role of mRNA. Okay, this is the role of mRNA. It carries amino acid to ribosome. This is the role of tRNA. Okay, then it is a constituent component of ribosome. This is a rRNA. Okay, and all of the above. All the statements are correct and this one is the correct option. Okay, all of the above is the correct option. Okay, what is next? RNA polymerase 2 is responsible for the transcription of. We have different RNAs, right? We have different RNAs to learn. Okay, we have different RNA polymerase to learn in the case of eukaryotes, right? Okay, so what are the different polymerase enzyme present? See, there are RNA polymerase 1. Okay, then RNA polymerase 2. Okay, then RNA polymerase 3. Okay, so we have learned this before, right? What is the role of these three polymerases? First polymerase, second polymerase will go. That is, it is the formation, it helps in the synthesis of mRNA. From hnRNA, what is formed? Heterogeneous RNA, mRNA is synthesized. Okay, so we can go with this option, that is hnRNA. Okay, hnRNA. Okay, what is the role of RNA polymerase 2? RNA polymerase 2 helps to synthesize the first RNA, that is heterogeneous RNA, which contains both exons and introns. Okay, clear? Is it clear for you? Is it clear? What is next question? If a person has VNTR, what is VNTR? Variable number tandem repeats. Okay, VNTR is variable number tandem repeats of 10 nucleotides in a satellite DNA of chromosome. In his satellite DNA of chromosome. Number 8, then what will be the VNTR of his son and daughter of that chromosome? Okay, so here only the father's VNTR nucleotides are given. Okay, the possibility of mothers is not mentioned, so we cannot predict it. Predict the number of VNTRs present in the daughter or the son. Okay, so what are the options given here? Both son and daughter will have the same. Son will have 8 but daughter will have 4. It can't be determined. Daughter will have 8 but son will have 4. What is the answer? It can be determined will be the answer. Okay, so what is the next question? What is the next question? A repressor protein is produced by, where you have heard about repressor protein? It is present in lactoperon, right? Lactoperon. So if you are looking into lactoperon, you can find different genes. Okay, you can find the gene for I. I is the repressor gene. Okay, then you can find ZYA genes. What is ZYA genes? Okay, ZYA genes. Okay, lac permease. Then YA for transacetylase. Okay, Y is for permease. Okay, so what is the role of these different genes you have learned in the lactoperon, right? So we can see, we can go to the question. What is the question given here? Repressor protein is produced. Repressor protein, what that is repressor protein? What is repressor protein? It is I genes. Okay, it is I genes and this repressor proteins is actually formed by which gene? Is that regulator gene, operator gene, structural gene or promoter gene? It is not the promoter gene. Okay, promoter gene is not doing any or it does not give any repressor protein. Okay, operator gene also does not give any kind of repressor protein. Okay, then structural genes, basically it is ZYA genes. It is also not giving the repressor protein. Then the only protein which is only gene which can give the repressor protein is regulator gene. Regulator gene. Which gene it is? Regulator gene. So we can go with option A. Okay, we can go with option A. So these are the different questions that is present in chapter number 5 MCQs. Okay, you can go through more MCQs. Okay, find these MCQs as important and go through the MCQ and do your best in your examination, score good marks. Okay, all the very best from the entire Go Score family. Score well. Thank you and have a nice day.